Hello, my name is Samreet Atwal, and my research is a potential mechanism for sensory perception to unpleasant florals and pear blossoms. So have you ever walked across campus and came across these particular set of trees in a winter quarter and it smelled really funky and you thought to yourself, what the heck is that smell? And then you talked to other students around campus and looked on social media, and then it turns out that people reported it smells like semen. Well, it turns out you're not alone in this regard. In fact, various other popular medical articles have reported on this. In fact, even the UCR Student Voice Committee have done it as well, and even the Highlander in 2015. So the pear blossoms, there are two of them. There's the Pyrus calicame, which are on the left, and the Pyrus caleriana, which are on the right. And first of all, we have to understand, how do we smell? Well, first off, we have odorants that bind to olfactory receptor neurons in the olfactory epithelium, and these olfactory receptor neurons project to glomeruli in the olfactory bulb. And the receptors are G-coupled protein-coupled receptors, and for each olfactory receptor neuron, there are one set for each. So it's not a mix and match of different GPCRs. And turns out that the glomeruli, each of them have a specific input from one type of ORN. So the result of this causes a specific spatial pattern of glomerular activity for each odorant. And the way we distinguish different odorants are from different combinations of ORNs being activated. So there are 5 questions we sought to answer with a literature review. One is what is the odorant responsible? What does it smell in other plants? Are the plant odorants similar to semen-like odorants? To explain association, the receptors involved, and lastly, why do we feel disgusted from the smell? First result showed not much in terms of the data. In fact, other Pyrus volatile organic compound analysis profiles only focus on leaves, not the blossoms. In fact, in here, none of these shown were reported to be the semen-like odor culprit. However, from other species, 5 of them, one Parling was shown to be the primary culprit. In fact, it very closely smells like human semen and possibly a vestigial pheromone. And the way this was done is through gas chromatography mass spectroscopy, which is a method in which a researcher can break down a smell to its individual components and see the percent composition of it. The known semen odorants are putrescine, spermidine, and spermine, and with one Parling, turns out there is a similarity. In fact, the structure of them all share an amine group. And of course, with amine group, they bind to these receptors called trace amine associated receptors, which are a subfamily of GPCRs. And there are 9 of them in the mammals. However, a third clade is not shown, which are teleos, which are ray-finned fishes like zebrafish. What's unique is that TAR1 is a non-olfactory role, whereas TARs 2 through 9 do have an olfactory role. Tertiary amines like trimethylamine, for instance, activate TAR5. Spermidine and spermidine, activate TAR9. However, putrescine has a possible mechanism to bind to TAR6 and 8, not confirmed. The way the receptor works is you have an odorant that binds. It causes G-alpha subunit to dissociate to bind to adenocyclase 3, which increases cyclic AMP levels. The cyclic AMP binds to cyclic nucleotide gated channels, which causes depolarization of the olfactory receptor neuron, and the depolarization is amplified via calcium gated chlorine channels. So the behavioral component involves the cortical amygdala, which plays a critical role in odor-driven behaviors. And particularly in my studies, it's shown that when you knock out these olfactory sensory neurons that sense aversive odorants, you lack the aversive response. But for learned associative aversive odorants, the behavior was intact. So in the olfactory bulb, you have a different set of glomeruli, one for learned and one for innate. In mice, it's shown that spermidine and spermine induce attractive behavior with TAR9. 2-methyl-1-parylene activates mice TAR5 to induce neutral behavior. And what's important to note is that 2-methyl-1-parylene has a structural similarity to 1-parylene, so there may be a possible mechanism of 1-parylene may bind to TAR5. And the combination of different odorants, aversive or attractive, can neutralize each other. Overall takeaway is that 1-parylene may be the primary culprit within the pear blossom set. The mechanisms are via TARs, and activation of these TARs induces aversive reaction to scent in humans. However, further analysis needs to be done to actually confirm the presence of 1-parylene in these pear blossoms. And once we know what the source of the odor is, we can play around with the olfactory system to, perhaps, like Jack Cole put it here, produce scentless ones. And here's the references. Thank you.